Hello everyone, Sangeeta Joyce here. Uh, Today's my topic for discussion is metabolism of cholesterol. Because of the structural complexity and the functional diversity makes the cholesterol as a fascination factor with almost all the disciplines in the modern medicine. Cholesterol has complex four-ring structure consists of a steroid nucleus having 19 carbon atoms, and we'll be discussing the cholesterol structure in detail. Together, the structure is known as cyclopentane phenanthrene ring system. Then for the biochemist, a simple 2-carbon molecule, 2-carbon acetate, with the help of 3 enzymatic reaction at 27 carbon, cholesterol can be synthesized. In case of physiologist and cell biologist, its essential function in membranes of animal cells. Then for physician, obviously, the clinical significance of cholesterol level, the elevated levels of cholesterol which accelerate the formation of atherosclerotic plaques leading to the heart attacks and strokes. Regarding the facts about cholesterol. Cholesterol, which is the major sterol in animal tissues. It is the precursor for other important steroids that includes bile acids, adrenocortical hormones, sex hormones, vitamin D, etc. and also cardiac glycosides. Next, no vegetable oil contains any cholesterol. There is no plant source for the cholesterol. Cholesterol is derived only from the animal food source. And the third fact, only a little portion of the body cholesterol is derived from diet. Remaining bulk of the cholesterol is synthesized from human body itself, that is known as de novo synthesis of cholesterol. And next, the bulk of it is synthesized in the body that I told, that is through the de novo synthesis. Let's move to the structure of cholesterol in detail. It has a ring structure named cyclopentanoperhydrophenanthrene ring structure system. That means all steroids have a similar cyclic nucleus resembling a phenanthrene rings, that is this structure A, B, C, that is known as phenanthrene rings. This attached to a cyclopentane ring, that is a fourth ring, that is known as cyclopentane ring. And the position of the carbon atoms on the steroid nucleides are numbered, that I will show in the coming figure. You can see from number 1 to total number 27. There are total 27 carbon atoms in the cholesterol structure, and it consists of a steroid nucleus having 19 carbon atoms. So up to here, this is total for 19 carbon atoms in the steroid nucleus itself. Then you can see that a methyl side chains which is attached as a single bond at the position number C10 and C13. C10 and C13. These are the two methyl side chains attached with a single bond at the position number C10 and C13. Then again you can see at the position number 17, C17, an 8-carbon alkyl side chain is attached to the D ring at the position number 17, that is C17. And also a hydroxyl group attached to the carbon number 3. Okay, this is all about the structure of cholesterol, which is known as cyclopentanoperhydrophenanthrene ring structure. Now coming to the functions of cholesterol. Most of the cholesterol in the body exists as cholesterol ester with a fatty acid attached to the hydroxyl group at C3. Then, secondly, it is a major structural constituent of the cell membranes of plasma, lipoprotein and also cell membrane. It is a major structural constituent in the cell membranes and plasma, lipoprotein. And next biologically important function is that cholesterol, which serves as a precursor for biologically important compounds such as steroid hormones, vitamin D, bile salts, etc. Okay, the steroid hormones are synthesized from cholesterol in the gonads and adrenal glands. And these steroid hormones are usually lipids, so that is why itself it can pass through the cell membrane. And it is divided mainly into, steroid hormones are divided into, like you know that it is corticosteroids and also sex steroids. Corticosteroids again divided into adrenocorticosteroids and glucocorticoids and mineralocorticoids. So glucocorticoids usually synthesized in adrenal cortex. Then sex hormones like for example androgen, estrogen, progesterone, etc., which is synthesized in the gonads and placenta from the cholesterol. So the major functions of being a part of the steroid hormones, the functions of cholesterol is indirectly it helps to control metabolism, then inflammatory reactions, immune reactions, then to maintain the water and electrolyte balance, etc. And also being a component of the sex hormones, it mainly helps for the development of sexual characteristics, and also it has multiple functions, like it has some ability to withstand the illness and injury. So these are the functions of steroid hormones. Now coming to the vitamin D. Vitamin D, which is derived from cholesterol and is a hormone which essentially helping to regulate the calcium and phosphate metabolism in human body. And coming to the bile salts and bile acid, these are also derivatives of cholesterol, act as a detergent in the intestine, helping to emulsify dietary fats to make them readily accessible to digestive enzymes. So all these are the functions of cholesterol. Okay, now let's move to the de novo synthesis of cholesterol or biosynthesis of cholesterol. So regarding the biosynthetic pathway of cholesterol is invented by the scientist Conrad Bloch in 1940, who has got Nobel Prize in 1964 regarding the same, and he has found out that the acetyl coenzyme, a 2-carbon molecule, is a source of all 27 carbon atoms of cholesterol. Now in detail regarding biosynthesis of cholesterol. Cholesterol is synthesized by a pathway that occurs in most of the cells present in the human body. About 1 gram of cholesterol is synthesized per day in adult, and I said almost all the cells can participate in the cholesterol synthesis. But the major organ in which cholesterol synthesis occurs in human body is mainly liver and intestine. Liver can contribute nearly 50% synthesis of cholesterol and intestine nearly 15%. And also I said almost all the cells can synthesize cholesterol. So the other major organs including skin, adrenal cortex and reproductive tissues. Now, regarding the enzymes involved in the synthesis of cholesterol, mainly the enzymes involved are present in cytoplasm and also in endoplasmic reticulum, that is the microsomal fraction of the cell. So if you are dividing the cholesterol biosynthesis into major 5 steps, the first two stages of cholesterol synthesis which occurs in cytoplasm, and the remaining 3 stages which occurs inside the endoplasmic reticulum. And the precursor for the cholesterol biosynthesis is acetyl-CoA, a 2-carbon compound, and it also requires the reducing equivalent, mainly NADPH, while ATP provides the energy for the synthesis of cholesterol. For production of 1 molecule of cholesterol, it requires 18 moles of acetyl coenzyme total and 36 molecules of ATP and 16 molecules of NADPH are required. Now the steps of cholesterol synthesis. It is divided mainly into 5 stages. The first one is formation of HMG-CoA and mevalonic acid. Second, conversion of mevalonic acid into activated isoprenoid units, which consist of 5 carbon units. Next, the polymerization of these 6 isoprenoid units to form a squalene. Then, next is cyclization of squalene to form the parental chiroid nucleus that is known as lanosterol, which is not shown here. Then conversion of lanosterol to cholesterol with the multi-enzymatic reactions. Okay, this pathway is crucial for the cholesterol biosynthesis. These first three enzymatic reactions, as per your syllabus, you are supposed to by heart these three steps only so far, but it is better to understand the remaining steps also. Okay, so let me discuss the first part, that is the synthesis of mevalonic acid from acetyl coenzyme. I said the first stage in cholesterol biosynthesis which occurs inside the cytoplasm. That is first two stages, that is up to the synthesis of mevalonic acid occurs inside the cytoplasm. First reaction, the first two molecules of acetyl coenzyme condenses to form acetoacetyl coenzyme, and the reaction is catalyzed by thiolase enzyme. So if you remember, last week I have taught you ketone body synthesis. In that also we have seen the same enzymatic reaction. The condensation of two molecules of acetyl coenzyme gives acetoacetyl coenzyme by thiolase enzyme, which was a mitochondrial enzyme, but here the thiolase enzyme and other enzymes which involving for the synthesis of mevalonic acids present in cytoplasm. That is the difference between these two stages. Next, acetoacetyl coenzyme condenses with one more molecule of acetyl coenzyme with the help of enzyme HMG coenzyme A synthase enzyme. Okay, and to form the intermediate that is known as beta hydroxy beta methyl. It is here written 3-hydroxy 3-methyl, that meaning beta hydroxy beta methyl glutaryl coenzyme, and the reaction is catalyzed by HMG CoA synthase enzyme. And the third reaction is the principal regulatory step of the cholesterol biosynthesis. Here HMG CoA is reduced by the enzyme HMG CoA reductase enzyme to form mevalonic acid. So the enzyme HMG CoA reductase helps to reduces HMG coenzyme A to mevalonic acid by the help of reducing bioequivalent NADPH. And this is the principal regulatory step in the cholesterol synthesis. Remember, HMG CoA reductase having importance in cholesterol biosynthesis because this is the regulatory enzyme for the cholesterol biosynthesis. And also, this is the site of action of the cholesterol lowering drugs. Many hypocholesтерolemic drugs, for example, statin drugs. You might have heard about statins while studying competitive inhibition. So, these drugs can inhibit HMG CoA reductase and inhibits the cholesterol biosynthesis. So, these steps are crucial for the cholesterol biosynthesis. Same pathway only, once again explaining the condensation of two molecules of acetyl coenzyme to form acetoacetyl CoA by thiolase enzyme, then synthesis of HMG CoA, then reduction of HMG CoA to mevalonic acid. Remember all these steps which occurs inside the cytoplasm. Now the second stage is which explaining the conversion of mevalonic acid to activated isoprene units. Isoprene units which consist of 5 carbon. Okay, so here the mevalonic acid is phosphorylated by ATP, the hydrolysis of ATP which gives the phosphate group to the mevalonic acid, and subsequently which undergoing decarboxylation to form the activated isoprene unit. So it is written several steps of phosphorylation and decarboxylation. Okay, so already we have explained around 30 enzymatic reactions are there for the synthesis of cholesterol. So here minimum 3 phosphorylation and decarboxylation occurs because they have shown the 3 ATP molecule and getting the synthesis of isoprenoid units like isopentenyl pyrophosphate. Okay, now the next reaction is that here you can see this IPP, isopentenyl pyrophosphate, can be interconverted to another compound, another 5-carbon compound, dimethyl allyl pyrophosphate. Okay, and these two can be combined to form another compound that is known as geranyl pyrophosphate. Here it is written geranyl, but that is a mistake only because it is a downloaded figure, that is why. The intermediate name is geranyl pyrophosphate. Next is polymerization of 6 isoprene units to form squalene. Before squalene formation, there are multiple reaction occurs. First of all, the condensation of IPP to IPP plus DPP, that is dimethyl allyl pyrophosphate and isopentenyl pyrophosphate, to give geranyl pyrophosphate, which is a 10 carbon compound. The enzyme catalyzing reaction is transferase enzyme with the elimination of inorganic pyrophosphate. Then 10 carbon geranyl pyrophosphate again condensed with one more isopentenyl pyrophosphate with the help of transferase enzyme to form a 15 carbon farnesyl pyrophosphate. In next, 2 15 carbon farnesyl pyrophosphate condensed to form squalene, and the enzyme catalyzing reaction is squalene synthetase enzyme. Here you can see the utilization of second reducing bioequivalent, that is NADPH to NADP. NADPH to NADP. Okay, so next is the synthesis of the squalene occurs here, that is 2 farnesyl pyrophosphate converted into squalene, a 30 carbon compound. And importance of the squalene is that it is the first isolated form of steroid ring structure. Okay, steroid ring structure. And why the name has given as squalene? Because it is first isolated from the liver of shark of genus Squalus. Squalus gene of liver sharks. Okay, that is why the name is given as squalene. And the next reaction is that cyclization of squalene to form parent steroid nucleus lanosterol. There was a small mistake. So squalene is not the first steroid nucleus. Lanosterol is the first steroid nucleus. So here squalene undergoing a series of complex enzymatic reactions. So many enzymatic reactions in presence of molecular oxygen and the reducing bioequivalent as NADPH, and multiple enzymes like epoxidase, hydroxylase, cyclase, etc. And till that it was a linear structure, and here till squalene it was a linear structure. Now it undergoing cyclization to form lanosterol, which has the 4 condensed rings that form the steroid nucleus of the cholesterol molecule. Okay, next reaction is that the formation of cholesterol from the lanosterol. This lanosterol is finally converted into cholesterol. Around 20 enzymatic reactions it is written. That means so many modifications of lanosterol steroid nucleotide ring structure occurs here, and cholesterol formation occurs. So the final compound of cholesterol should contain 27 carbon atoms. So the changes might be removal of 3 methyl group at a C4, then the shortening of the carbon chain from 30 to 27 by removing 3 carbon atom, and second, removal of 3 methyl groups, extra methyl group at a C4, then migration of the double bond from C8 to C5, then reduction of the one double bond between C24 and C25 by NADPH. So all these reactions leads to the synthesis of cholesterol from the lanosterol. Okay, so this is the overall pathway of the cholesterol biosynthesis. My advice is that please go through with the textbook figure. This figure is not from the Vasudevan textbook. Better you can take the figure from the Vasudevan itself. Okay, so once again, synthesis of mavulinic acid. Up to mavulinic acid, that is stage 1 is very crucial. You have to by heart those stages, because most of the application of cholesterol biosynthesis is mainly from these steps only, up to the synthesis of mavulinic acid. Remember, the rate limiting step is that HMG-CoA reductase enzyme, which can be inhibited by various factors like various drugs or by feedback inhibition, cholesterol itself or mavulinic acid. All these can inhibit the HMG-CoA reductase enzyme and can regulate the cholesterol biosynthesis. Okay, and further we have seen so many enzymatic reaction, phosphorylation, decarboxylation to form isoprenoid unit, that is a 5 carbon compound, then 2 isoprenoid unit to form 10 carbon geranyl pyrophosphate, plus one more isoprene unit to form 15 carbon farnesyl pyrophosphate. Then so many enzymatic reactions leads to the formation of squalene. Then squalene undergoing cyclization to form lanosterol. Lanosterol is the first steroid molecule before synthesizing cholesterol. Then with several, nearly 20 enzymatic reactions, leads to the modification of the 30 carbon lanosterol to finally a 27 carbon cholesterol molecule. Okay, so this is all about the synthesis of cholesterol. Now regarding the regulation of cholesterol biosynthesis. HMG-CoA reductase, I already explained which is a rate limiting enzyme in cholesterol biosynthesis, and there are three different kind of metabolic control are there with this enzyme, that is feedback regulation, then hormonal regulation, and the third one sterol mediated regulation of transcription. So let us see one by one. Okay, here by seeing the picture itself you can understand cholesterol biosynthesis is already a complex one, and how it is regulated. Okay, so it is mainly regulated by the intracellular cholesterol concentration and the supply of ATP and the hormones insulin and glucagon. Okay, so let us focus on the HMG-CoA reductase enzyme. That is a major regulatory point which helps for the conversion of HMG-CoA to mevellinic acid. Okay, now there are different type of regulation of cholesterol biosynthesis, that is short-term regulation and the long-term regulation. Or first let us explain the covalent modification or short-term regulation, the activity of existing HMG-CoA reductase. It is regulated by the reversible covalent alteration. You can see in this figure that HMG-CoA reductase active and inactive. Okay, so what is the meaning of active and inactive here? This figure you can see that HMG-CoA to phosphorylation. That means phosphorylation and dephosphorylation is the point for the reversible covalent alteration. So the phosphorylation and dephosphorylation, it is occurred by usually AMP dependent protein kinase. Before that I have to say the hormones glucagon which stimulate phosphorylation. Like here you can see glucagon which stimulate phosphorylation. So phosphorylated HMG-CoA reductase is actually inactive. So what does it mean? Glucagon which inhibits the cholesterol biosynthesis. Next, insulin. Insulin which promotes the dephosphorylation. You can see insulin catalyzing the protein phosphatase enzyme and which causes the dephosphorylation of HMG-CoA reductase. So the dephosphorylated form of HMG-CoA is actually active. That is why the hormone insulin favoring the cholesterol biosynthesis. So this is regarding the hormonal regulation. Okay, now coming to the another important factor which I have not shown in this figure, that is phosphorylation and dephosphorylation occur by AMP dependent protein kinase, that is AMPK enzyme. This usually senses the high AMP concentration. What does the meaning of high AMP concentration? That means low ATP level. So when the ATP levels are dropped, what will happen? AMP dependent protein kinase will be getting activated, and that also causes the phosphorylation and inactivating the HMG-CoA reductase. So thereby the synthesis of cholesterol is inhibited in the low energy status. Okay, and another regulatory step is long-term regulation. Here the multiple number of molecules of HMG-CoA reductase is increased or decreased in response to the cellular concentration of cholesterol. Okay, second point I was discussing, intracellular cholesterol concentration also can regulate the cholesterol biosynthesis. Okay, here HMG-CoA reductase in liver is inhibited by mevellinic acid itself. You can see here mevellinic acid itself can inhibit the cholesterol biosynthesis. Okay. Then regulation of HMG-CoA reductase synthesis by mevalonate and cholesterol is mediated by another important mechanism that is known as transcriptional regulation of HMG-CoA gene. Okay, that is known as sterol mediated regulation of transcription. So the increase in intracellular cholesterol can cause decrease the transcription of HMG-CoA reductase gene. Okay, so mevalonate and cholesterol represses the transcription of HMG-CoA reductase via activation of transcription factors, that is specifically sterol regulatory element binding protein, thereby which decreases the cholesterol synthesis. So increase in cholesterol inhibit the activity and synthesis of HMG-CoA reductase indirectly by decreasing the transcription of HMG-CoA reductase enzyme and decreasing the cholesterol biosynthesis. And another important inhibitory factor is that hypocholesterolemic drugs, like specific drugs which can inhibit the HMG-CoA reductase enzyme, which includes the statin group. Example, pravastatin, lovastatin, mevastatin, simvastatin, fluvastatin, etc. These are the brand names actually. You should remember the statin drugs which inhibits the HMG-CoA reductase enzyme. So these statins are usually the structural analogues of intermediates in the conversion of HMG-CoA to mevalinic acid. Here, which explain the competitive inhibition. Statins having structural similarity with the HMG-CoA, so both will compete with the enzyme HMG-CoA reductase, thereby it inhibits the conversion of HMG-CoA to mevalinic acid, thereby which can decreases the production of cholesterol. So this is all about the synthesis of cholesterol biosynthesis. Part 2 also there, in which I will be explaining the metabolic fate of cholesterol, or which can explain the biologically important compounds which is synthesized from cholesterol. So kindly watch the second video also, that is which explains the metabolic fate of cholesterol. Thank you.